viewers, we want to look on bacterial infections, a small concept in terms of classification and management. Today we will look at classification based on gram staining. So what is a bacteria? We can start by defining what a bacteria is, and we can say it is a microscopic single-celled prokaryotic organism which is found everywhere. It can be in the soil, in the water, in the environment. And what is the bacterial infection in this case? A bacterial infection occurs when a harmful bacteria enters the body, multiply and causes damage to tissues and organs. So you can see the bacteria are classified into two based on the gram staining. You can have a larger group of what we call gram-positive bacteria. You can also have gram-negative bacteria. So that is the classification. We have various types of classification. We can base on the shape of the bacteria. We can base on the gram staining, as we have said. You can also base the classification on oxygen requirements, nutrition, other properties such as spore-forming and non-spore-forming bacteria, motile and non-motile bacteria. When we talk of gram-positive and gram-negative bacteria, we are basing on gram staining. In this case, gram staining is a laboratory technique which is used to classify bacteria into two major groups. In this case, gram-positive and gram-negative. So what is the importance of classification? One, we want to look at rapid diagnosis, and also it guides antibiotic therapy. So this is the steps for gram staining. You prepare the slide, you do what you call primary staining, you flood with iodine, you decolorize with alcohol, you counterstain with safranin, and then you do microscopic examination. And what you need to remember that after every staining, you stain for 1 minute, and then rinse with water, stain 1 minute, rinse with water, until this stage. So what are the results? When you see a purple bacteria or a purple color, that means that's a gram-positive bacteria. When you see a pink or red color on microscopic examination, that is a gram-negative bacteria. Now, these are the similarities and differences between gram-positives and gram-negative bacteria. When you look at a gram-negative bacteria in terms of structure, you will see that the internal structures of the bacteria are similar, but the difference comes on the cell wall. When you look at the gram-negative bacteria cell wall, you will find that it has an extra layer, what we are calling the outer membrane of lipopolysaccharides. When you examine these two bacteria carefully, you will realize that this bacteria lacks this outer wall. And this outer wall is very significant because it has effects on management of these bacteria. You will realize that in the management of gram-negative bacteria, they are slightly stubborn compared to gram-positive bacteria because of this extra layer. This extra layer offers what we call barrier mechanisms of these bacteria. That is why when we are selecting antibiotics for treatment of these bacteria, the gram staining technique is very important. You need to know, are you dealing with the gram-negative bacteria or gram-positive bacteria? Therefore, this layer here, it means when you are selecting the antibiotics for management of this bacteria, in the event you are dealing with gram-negative, you will require antibiotics that are very special to penetrate this outer layer because it offers what we call barrier effects. So what is the nature of the antibiotics required in this case? You will need antibiotics which have very, very small molecular structures to enable them to penetrate through this layer. This layer you see around here as what we call porins. Porins are what we call protein channels that can enable very small molecule very small molecular antibiotics to penetrate through. Therefore, we need antibiotics that have very small molecules, and therefore that antibiotic should also be what we call hydrophilic for easy penetration. Thank you, and next time let's see, we'll talk about action potential.